What is the TMS-EEG experiment, and can it distinguish conscious from unconscious people? So TMS-EEG experiments on the various levels of consciousness has been already done. And they did through this, stimulating the brain, bypassing sensory and motor pathways, and then that addresses the issue of the stimulus and the report kind of paradigm. Even without stimulus, even without the reports, people are conscious and so on. And so bypassing sensory and motor pathways, we may be able to directly probe consciousness per se. And then it can be applied to those people who have problems in sensory motor pathways or verbal reports. So, people who doesn't have any, let's say have a problem in the ear, they may be missed in terms of, presence of consciousness if the communication to make sure what they are conscious is done through the sort of the, auditory communication. But this one directly bypasses the sensory stimulus. So it may not be a problem for them. And also it doesn't require verbal reports. So even it can be, applied to the not only locked-in syndrome patients, but also newborn babies or whatever, even animals as well. So the idea is to measure the both responses and then compute its complexity, which reflects both information, differentiation, and integration. The currently most promising measure of consciousness is regarded as a perturbational complexity index, or PCI for short. And this has been extensively studied by Marcello Massimini in the photo here. And he has written a book with Giulio Tononi, the inventor of the integrated information theory of consciousness, from Oxford, Sizing Up Consciousness. And I think it's quite interesting and highly readable account of consciousness, so I recommend it. So what Marcello and Giulio did is the following kind of experiment. So this is a page participant on the left side, and they wear this EEG cap. You see a lot of connected lines to each of the electrodes that measure the brain wave, and HD means high density EEG. And then this device, gigantic device, is TMS, transcranial magnetic stimulation. And then this is a neurobehavioral system that precisely monitors the angle and the distance and the location of the TMS with respect to the brain and the EEG. And that is important to do this experiment. So transcranial magnetic stimulation is idea to use this electromagnetism. If you pass the electricity through this kind of coil, one way, one direction, really strongly, then that generates the induced magnetic field. And then induced magnetic field eventually, induces electricity in the surface of the brain or deeper in the tissue. And then it's known to excite or stimulate, inhibit the neurons underneath the coil. And that's the transcranial magnetic stimulation paradigm's idea. So that advantage of this method is that it can skip all these the processing from I to LGN to B1, and also ear to auditory cortex, which may be not really related to consciousness per se, but it is just used as a sort of conduit for the the central processing. And as I said, the many of the locked-in syndromes people don't have this possibility to communicate through the sensory input, and so directly probing or stimulating this brain is one important point, which can generalize across the people without intact brain mechanism across all the sensory modalities. All right, so the first of the Marcello's study used this system, and then to locate the TMS at the front of the brain, like depicted in here. And then there were 60 electrodes, and then over the time, they measured the response. Okay, and then here, what's shown here is the each of the electrodes is, going from one to many electrodes to the bottom. And then, so this is roughly 68 electrodes and so on. So each line of the black thing is basically the sort of voltage fluctuation of each electrode. And then the TMS pulse is provided at this point or this point. And as you may notice that when the subjects are wakeful, then stimulating a brain at this location doesn't cause a huge response. And it seems like it's almost the same as the ongoing, sort of the background activity right here. But there is something that is actually induced by this stimulation here. So that's what happens when TMS is provided when people are awake. And in this case, they didn't feel difference in terms of conscious sensation. And then under the different situation of the conscious states, when they did that with a non-REM sleep, where only 50% or less time of the people experience anything in terms of the dream. So under this situation, if you provide TMS stimulation here, then you sometimes see this big fluctuation, like here, induced fluctuation, but it may not be related to TMS itself because you didn't see it here. But somewhere around here, there is some kind of component that is reliably and strongly induced across the electrodes. So it seems like similar to this non-differentiated, non-informative kind of situation. And we can try to see it later more clearly. So the idea here is that then exactly the same location is stimulated and recorded with the EEG under both wakefulness and also non-REM sleep. And here is the EEG response of one particular electrode. Okay, so here this is called event-related potentials because event is now defined as a TMS. Okay, and then these are 100, 200 to 300 milliseconds before the TMS pulse, and then the right side is after the TMS pulse. And then what it means is that and this is going from trial 1 to trial 450 along the y-axis. The color of this figure indicates the voltage that is evoked by the stimulation, and it's smooth across several trials, maybe tens of trials or so. And then so each particular point is the average across trials along y-axis. And what you can see here is that initially around this up to here, the evoked response is relatively small here, especially here. And then, but it continues. On the other hand, as the time goes by, you start to see that the initial response becomes much, much bigger. And then kind of becomes silent over there afterwards. And here's the mean response within this period where they found or they confirmed the wakefulness of the subject. And then that shows this initial complicated kind of pattern of the response. And on the other hand, the stage 1 sleep, which is associated with some type of a bizarre dream and so on. There is some kind of complex pattern, but more importantly, non-REM sleep afterwards is characterized as an initial really big response. By the way, this is scale that is consistent across a stage that leads to silencing afterwards. Okay, so that is consistent with what we thought would happen for the integrated, too much integration and the less informative system. And here's another type of the representation of data. So what you're seeing here is over this time scale. So this is roughly 30 milliseconds after the TMS onset. And this is already averaged across all many trials. And then it goes from 0 to roughly like 500 or 300 milliseconds after the TMS. And each of the line corresponds to each channel's response mean over the trial. So what I showed in the last figure is one particular channel's response, which corresponds to something like, this thing. Okay. But now all the channels, 60 channels, are presented at the same time. And that's why you see lots of lines here at the same time. And then if we look at the response at the one particular time point, let's say 10 milliseconds at here, then across the brain areas. So this is representing all the brain electrodes. And then at the surface EEG level, if you see some kind of positivity here, and that corresponds to maybe positivity around these red electrons, and the negative ones are the ones that are suppressed or going down around the time location of the TMS. And you can make some kind of a snapshot like this over the time scale, 20 milliseconds, 51, 63, 83, and so on at each peak of the red part. And the red part is the one that is above or below the baseline period here. So if it basically measures how much the TMS evoked the response above and beyond what is expected from the background activity. And then what they did was to do the so-called source localization estimation. Using some kind of brain modeling, they could locate where this activity is coming from. And the unit is called current density response. And here, they make some kind of three-dimensional modeling of the brain and then locate where the activity is coming from. And that is better than just looking at the EEG surface amplitude. In this case, they found that, okay, so initially after the stimulation of the, this part, that part itself becomes activated and it propagates to, both front and also different area, other side of the brain, up to, here, left side of the brain. And then also going backwards, parietal areas, and then coming back to the left, and then to right as well. So it moves, the focal area of the brain activation moves over the time. And that is consistent with the prediction of this integrated information model of the 8 neurons. When each of them are differentiated and have a unique functionality, stimulation of one area should propagate across all the areas and then coming back. Now, when they did the same experiment with the non-REM sleep, where people don't have much complex experience, and when woken up, only 50% of people report dreaming of the simple kind. That's this type of the response. So initial response becomes much bigger. In fact, here and then here, this is actually bigger than the magnitude that you observe in the wakefulness. But then afterwards, initially there is some kind of uniform activation that is observed around the area of the stimulation. You do see the activation across many areas, but when you do source localization, it turns out that response is remaining actually at the same location, more or less, and then confined within that areas and then dies out relatively quickly. So this is, in fact, it's similar to the situation of the disconnected, non-integrated loss of information type of the 8 neuron system. Two neurons connected to each other and it doesn't get affected other pairs. And this is a schematic of many of the types of experiments that Marcello Masmini and Antononi's group did. Under the wakefulness, the TMS produces many different kind of complicated patterns. On the other hand, non-REM sleep, some people produces and also some voltage of the stimulation, like here, weak voltage of a TMS, like 90 volts per meter. Then it results in this, very local activation. Or if it's very strong, 160, then it can evoke the response across all the areas, but it dies out together. And then other types of the sleep, REM sleep, which generates conscious experience of the dream. And then that tends to make a response that is very similar to wakefulness. So the evoked response becomes complex and also propagates and coming back and so on. On the other hand, general anesthesia that knocks you out in terms of consciousness, that shows very similar kind of pattern as a non-REM sleep. It's either local or doesn't propagate across doesn't propagate across the brain, or propagates too much and then uniformly dies out. And then the most sort of the clinically relevant is this, different kinds of the brain injury. Vegetative state or wakefulness syndrome, they are similar to general anesthesia or non-REM sleep. It either shows a focal pattern or uniformly excited and then dies out. Minimally conscious patients, such as those who can actually imagine things and make some kind of meaningful response, although from outside it's almost indistinguishable from the vegetative state patient. They are like a wakeful patient. And then locked-in syndrome patients who are able to make communication with the eyes and the eye movements and blinks and so on, they are, of course, similar to wakefulness patients. So the distinction between this vegetative state versus minimally conscious state is difficult. Locked-in syndrome is easier because they can actually make voluntary eye movements. But here, the potential is that the minimally conscious patients can be distinguished from the vegetative state patients. So in summary, TMS evoked EEG patterns are already different depending on the brain states. The highly integrated information pattern is correlated, most likely, with more complex TMS-EEG patterns. The low integrated information should correspond to low complex, much less complex TMS-EEG. And in fact, this type of the result has been confirmed across different types of the loss of consciousness, going from anesthesia, sleep, and also brain lesions. And based on this data and the result, can we construct a measure of consciousness, objective one, like a consciousness meter? That's the next question in the video.